so yeah, we have a lot of projects going on. I'll overview quickly the different ones, and then I'll talk at the end about the strawberry guava project and where that's at. Um, let's see if I can advance. So unfortunately, there's a lot of work for me to do. We have so many really important weed problems. And so this here's a list of some of them. Um, and so, yeah, I'm really motivated by the need of natural resource managers in Hawaii for more tools. And so the idea is that biocontrol using natural enemies that are really highly specific is one important tool that we need for a lot of these very impactful weeds. So our process, we do all the different stages of biocontrol work, from selecting targets in consultation with natural resource managers, and then exploring for the natural enemies in the countries where the weeds come from. If we find something good, we'll bring it back to Hawaii and test it in a laboratory setting against native species, and learn as much as we can about it in containment. We are also studying them intensively in most cases in the foreign countries where they occur naturally. And then finally, if we can get, if the data show that the thing is specific and worth releasing, then we apply and get permission, and then eventually, if possible, release and monitor the impacts. So we're doing all of these different projects, and they're all at different stages of this process. So one of our insects is on the docket for release in Hawaii right now. It's in the review process. Is Euchalazia chrysope. So it's this butterfly that feeds on Myconia leaves, and the larvae feed in these impressive groups and follow each other around as a family group. It's a very interesting biology. So this is an insect from Costa Rica that we've tested a lot, and we know that it's the only thing in Hawaii that it's going to eat is Myconia and one other melastome, Tetrazygia, which is now also called Myconia species. So yeah, it's very specific and hopefully can do a significant amount of damage to the leaves of Myconia. We're also working on other agents as well, especially some things that attack reproduction. There's also still there's been one agent released in the 90s, fungus. There's another fungus that occurs throughout the native range that could be really good, but unfortunately we don't have a facility right now for doing pathogen work in Hawaii. So that's a big challenge for us right now in getting work done on certain types of agents. But that could be a good one for the future. The other insect that is currently under review is this beetle, Cyphrea uberabensis. So this is a little Brazilian beetle that feeds on Tibicina, and also it turns out it feeds on some other melastomes as well. So these data show, this is kind of a typical sort of data set that we do with our research, feeding different plants to the insect where they only have one choice. They're put in a cage, they just have one thing that they could possibly eat, and we measure whether they eat it or not. And these things eat melastomes, and specifically a variety of melastomes in one tribe of that family of plants. So they're quite specific in both their feeding and their egg laying. And one thing I should point out is people often say, oh, but they're feeding a little bit on some other things that aren't related to mellostomes. And that's very normal in this kind of testing, that when you put an insect in a cage and you don't let it do anything, it'll take a few bites out of something. It might even lay an egg or two. That's kind of a normal behavior, and it doesn't reflect anything that happens in the real world, because they're very specific. If they're on something that they don't like in the real world, they leave it and go find what they like. So anyway, this kind of shows some of the data for these insects, which hopefully we can get through the process and be releasing sometime in the next year, for control of some mellostomes. There's been a history of work on Clydemia, and with a variety of agents that have been released in the past. None of them have really done a stellar job. So we definitely need something more. And one of the things I'm working on is a wasp that lays its eggs and creates galls in the fruit. There's also a nematode, a type of pathogen that makes galls in all kinds of the new growth of the plant, that I would love to advance. That's a challenge, though. We don't have a facility for that right now in Hawaii. So we're doing some of this work in other places. This is one we've got bugs in the quarantine lab right now. Chromalina, the devilweed, which has been a problem on Oahu for years, and is now on the Big Island too. So this fly lays its eggs in the new growth and makes galls in the stems. So we're just figuring out how to rear it and starting our testing this year. So it's been used elsewhere around the world. We got it from Australia, where they've started using it. It's been used in Guam for years, India, Papua New Guinea, different places. So it's already been tested a lot. So we just have to kind of do a little bit more testing of certain Hawaiian plants, and then we should be able to have the data that's required to propose release. Big project. After Myconia, the Myconia project, this is my new biggest project. Most recent is Albizia. And I'm assuming you all know how bad Albizia is in our native environments. It really transforms the environment. And it's bad for native species like Metrosideros. So we've been working for the last 5 or 6 years with colleagues in Indonesia and Papua New Guinea. We started a partnership with folks in New Zealand now, because they do biocontrol research too, and Australia. So we've got a good group of partners now working. We've identified a few potential agents that we're kind of delving into in more detail, but it's still early days for these ones. We don't know which ones are going to prove specific and that look like they'd be the most impactful. So these projects take a long time, and we're kind of still in the beginning stages for this one. Just so you all know, Albizia comes from Papua New Guinea and Indonesia. Those are its native countries. So that's where we're going to work for this. Rubus is a very nasty weed, Rubus ellipticus in particular. We have a collaborator in the United Kingdom that does biocontrol research around the world, CABI. And so they've been working with us off and on for several years, exploring mainly in India now for natural enemies. So this again is still in that exploratory phase. There have been some natural enemies that look like they're worth looking into more. Pandemics are not good for international work. So this project has really been on hiatus kind of for the last bit, but they're gearing up to get back into India and trying to look at some of these agents some more. They also work on Rubus niveus for us and for it's a big problem in the Galapagos. So hopefully they can find something or multiple agents that work for both of these pests. That same collaborator, Cabby, also has a project working with us and the New Zealanders to find natural enemies for ginger. In particular, for us, Kahili ginger. And this fly is the one that I kind of have the highest hopes for. We don't know yet if it's going to be specific enough. It may be that it eats other gingers in the Hedychium genus, which would include some plants that might have some horticultural value in Hawaii. So there's going to have to be some discussions about what we want in ginger biocontrol. But yeah, we're still seeing what is out there and what we might use with partners. Probably, most likely, the New Zealanders will be getting the first experience in terms of releasing agents against these gingers. Phaya tree, formerly Myricophyia, now Morella phaya, has been a concern, especially on the Big Island, Maui, Kauai also. And we worked on it, my predecessors worked on it in the 80s and 90s. And there were some agents released, but nothing that's been impactful. So we've gone back to start to try to look at for natural enemies for it. And this weevil is one possibility. We really don't know for sure what holds the most promise, but there's good collaborators and potential for investing in this project. It basically hasn't been started. It's been exploratory, preliminary exploratory work at this point. But this is one project I'd like to see advance before I retire. This agent on Brazilian pepper tree, also known for us as Christmas berry. This has been released in Florida. They're looking to release it in Texas. It's been tested against Hawaiian plants, and the data are still need to be evaluated in terms of how specific it is. But it's one of the agents that has potential for a Christmas berry. So we're still interested in kind of evaluating it. This is why it takes years, because testing gets done in one place, and then we might need to do additional testing here in Hawaii. So yeah, there also can be concerns about kind of competing interests, whether Christmas berry is a weed that really needs control in in the natural areas and everywhere it's invading the ranch lands. But it's also a resource that honey producers use. So there's more discussion needed there. I'm doing okay time-wise, yeah. This is another kind of potential project that's been developed elsewhere, but I haven't pursued it yet. But there are known natural enemies that are specific to African tulip that we could potentially use in Hawaii. Another project that really hasn't been advanced far at all. Potentially looking at natural enemies for fountain grass. The White Department bag has done very preliminary work, but this is another one that is important in the state and could be looked at. And more other partners around the world are looking at grass biocontrol. So this is kind of, there's potential for work here. Okay, so quickly I'm going to go through about where Strawberry Guava project is. It's been released all around the state. We've studied it in in various experimental plots on the Big Island intensively. We're finding that after several years, it starts to slow down the growth of the trees, but it does take several years. It's gradual. Also, it's starting to show its ability to reduce fruit production very gradually. And yeah, especially on the common red and yellow varieties, not so much on the spindle variety, which is more of a problem on Kauai and Oahu. And we've released it, some of the sites, the earliest site we released it on the Big Island, it's spread from that site several kilometers as far as we know on its own. So it is able to spread with the wind. And it's a pretty fair distance, on average 400 meters a year from this site, if in fact it's all self-spread. So that's promising, but we are going to have to help it along if we want to get it deep into remote forests. So we're working on that in terms of how to drop it from drones and helicopters. So yeah, that's where that project is right now. And that's it.
So that's my fruit fly head, which I had a student who was working in the museum before, a very talented artist, in addition to entomology students. So I had it custom made, and it's all anatomically correct. And I use that when I do outreach. So it's for outreach and extensions, it's really great. So forgive my voice, it's not really great. I'm coming out of a cold, so my voice will be normal again, but I was a classmate with Roshan for many years, so I was very happy when Roshan, when you invited me for the talk. So let me share the screen. Yep. Share. Do you see it?
Yes, yes.
Hang on. You still
Full
see
screen.
it?
Yes,
Huh?
but we need full screen.
Okay. So is it full screen now?
Not yet.
How about now?
No, not yet. Now, yes.
Okay.
Yes, yeah.
Okay, so I'm going to talk about the various eradication programs against invasive fruit flies that went on over the decades in the Pacific Islands. So in Oceania, including Hawaii, there are 44 species of pest design fruit flies. You have 5 of them in Hawaii, and some of them have invaded the South Pacific Islands. And all the invasive pests, there have been attempts, successful or not, to eradicate them. So 5 species of invasive fruit flies were targeted in eradication programs over the years. So you get the map here, Oriental fruit fly, melon fly, and Mediterranean fruit flies, which are have been introduced to Hawaii, and Queensland fruit fly, native to Australia, and Bactrocera xanthodis, native to the Polynesian islands. And some of these programs were successful, others did not work out. And we're going to see the reasons why there was success and in other cases, no success. So the technology used to eradicate fruit flies, well, all of you are residents of Hawaii, so you're all familiar with fruit flies. We have the male lures, the methyluginol and Q lure, which we use in traps to monitor and control fruit flies. You can apply a protein bait solution to attract with a bit of pesticides to attract female fruit flies. So you have a tool for males, an attractant for females. You can also breed in lab and release sterile insects, sterile males that will breed with the females that will lay sterile eggs. And crop sanitation, all the fruits at the bottom of orchards, destruction of these fruits will eliminate breeding sites of the fruit flies, such as in this orchard here in French Polynesia. And he's a Tahitian guy, a former colleague here doing the bait spraying. So the very first attempt that was a success to eradicate fruit flies was actually in the Mariana Islands. So Oriental fruit fly was eradicated from Guam and Northern Mariana Islands using a combinations of male annihilation and sterile male releases. And they were the Oriental fruit fly through releasing from airplanes, these blocks dipped in NALED and methyluginol. It was eradicated in 6 months on Rota, 4 months on Guam. So Rota through male annihilation, 6 months. Guam through sterile insect release, it was eradicated in 4 months. And what helped the eradication is that there were 2 typhoons in 1962, just before the eradication. So the typhoons reduced the fly populations to a very low level. That's why it was so easy to eradicate. And from the northern, the CNMI, it took the sterile insect release were not enough to eradicate. So they had to complement it with male annihilation. So it took a couple of years to eradicate. And the person who implemented that program was Ernest Harris, who joined USDA ARS in Honolulu in 1962. And he was from Arkansas, and Ernie was one of the very first African-American entomologists, researcher in USDA ARS. I used to know him very well. He was, and I'm sure several of you have known or worked with him. So he's a legend in Hawaii. So melon fly also was eradicated through sterile insect technique. So the sterile flies were bred in Honolulu, and the pupae were sterilized with radiation and then sent by plane to Guam. And the flies were emerged from pupae in release boxes that were dropped from airplanes and dropped on the ground and ground release cages. So again, in a couple of years, they were able to eradicate melon fly to 2 major pest species eradicated from the Mariana Islands. So melon fly from Northern Mariana Islands, they were going to eradicate from Guam, but the program was terminated such that melon fly was reintroduced from Guam to the Northern Mariana Islands in the 1980s. And ivy gourd is the major host that sustains very large populations in the Mariana Islands. There's potential for area-wide control of melon fly in the Marianne and Guam and CNMI, and growers are applying control, but they still have a long way to go to really have a fine-tuned program. And I'd like to work with their entomologists in the future on that. So in Cairns, Northern Australia, Oriental fruit fly was detected in October 1995. And through male annihilation and protein bait sprays, they were able to eradicate. So the month after it was detected, they initiated an eradication program, and they were able to eradicate it in 1997. So Oriental fruit fly introduced to New Guinea in the late 90s, and that's how it was introduced to Cairns. But it took, they believe it had been established 2 or 3 years earlier and was unnoticed. So the program had to be over a very large scale. Had they had a network of surveillance traps to detect for early detection, they could have detected and eradicated a lot earlier, but government at that time decided to save $20,000 of budget to discontinue the trapping network. So there they paid the price. So they did the eradication through male annihilation with sugarcane block dipped in malathion and methylogenol, and malathion and protein hydrolysate. So the last fly was trapped in late 1997, and 6 months later, they declared area freedom so that they could start trading the fruits produced in northern Queensland. So the eradication cost 33 million Australian dollars. Sounds like a lot, but if you take into consideration the benefits, if they had not eradicated it, they would have lost permanently export of some of their fruits unless they have post-harvest treatment. And the additional cost without eradication would have been $30 to $40 million. So $8 million a year. And the trade losses, in 1995, they lost almost 3 million of overseas trade. But within Australia, the temporary ban on banana export to other parts of Australia, they lost $200 million. So the cost versus the benefits is pretty obvious here. And that was published in this paper, this book that has very good socioeconomic studies on eradicating fruit flies. So in 1996, the people from the regional fruit fly projects in the Pacific, out of which I was, here I am here with a big beard at that time. So we all got together in 1996 to Cairns and received training on eradication of fruit flies to see how it's done. It was 1 week, and the emphasis was on prevention. Try to establish a surveillance network for invasive pests in our countries. I was working in the Federated States of Micronesia at that time. I represented that country in the conference. And preparedness, formulation of emergency response plans to deal with fruit fly invasions, and public support, awareness, public support for fruit fly eradications in case they invade countries. So the original fruit fly project was initiated in 1990, and I joined it in 1994. Some of the activities were to promote alternatives to pesticide cover sprays like protein bait sprays, fruit bagging, and even the local productions of protein bait This is a bait unit, production unit in Tonga, and enhanced trade by studying the heat tolerance, breeding in lab and studying the heat tolerance of the pest fruit flies in the Pacific, and developing heat treatment units that are now in 6 different countries that allow to export fresh fruits, heat treated, post-harvest treatment done by the Pacific Islanders. So I was talking about quarantine surveillance for early detections of invasive fruit flies. Should a species from Solomon Islands be introduced to Fiji, there's network of traps to quickly detect it and have a quick response to eradicate invasive fruit flies. So there's a network of traps all over the Pacific Island countries that was established. So we'll go back to that network. New Zealand, they have pretty much the best fruit fly early warning and surveillance and quarantine inspection system in the world. They're really the leaders. As a result, they detected a Mediterranean fruit fly in Auckland in May 1996. And because of very early detections, they were able to eradicate it in 6 weeks. The benefits of preparedness. So the original fruit fly project, we all went to cairns and saw an eradication in progress, but the project wanted to have an actual hands-on exercise of actual eradication somewhere in the Pacific. And the island of Nauru in Micronesia had 4 fruit fly species. 1 native mango fly and the 3 others, these ones introduced from Taiwan, and Pacific fruit fly introduced from Fiji. So the original fruit fly project established a pilot program to try to eradicate 4 species of fruit flies. And condition for that program would be that Nauru establish a quarantine inspection service, which they didn't have. So for eradication, and I did like half a dozen trips to Nauru to help because I was part of that team that did the eradication. So we dipped canite blocks in male lure mixed with fipronil, first use of fipronil for eradication, nailed them to trees every 2 months, and did some limited protein bait sprays. So the protein bait that you have in Hawaii, you spray it on leaves, you lose a lot of it through rain. So it has its issues. So in Nauru, we used the fipronil plus protein mixed with an additive that forms a gel, hence Bactrogel, and Bactrogel adheres to tree leaves and resists a lot better the pouring rains. So that would be really well adapted for a Hawaii environment if ever it becomes registered for use by growers. So in that program, the Nauruan community was actively involved. So this is the youth group. These are high school children, and these are the phosphate industry worker, because Nauru was a phosphate island, and some more workers from Kiribati, and I worked with that team, really great people. So as a result, Oriental fruit fly was eradicated in 2.5 months with male annihilation blocks. Melon fly with male annihilation and 1 application of protein bait eradicated in 2.5 months. Just like in the Mariana Islands, the typhoon reduced the fruit fly populations. Here, In Nauru, they had a couple of years of drought before the eradication, and the drought reduced the host plants. So the melon flies were in very small numbers, so we were able to knock it very quickly. Pacific fruit fly took 17 months to eradicate. The reason is methyl is not a powerful attractant, but more recently in 2017, 2018, an alternative to methyl was discovered that's a lot more powerful. And I tested it in Wallis and Futuna 3 years ago in my last trip to the Pacific. And it works really well. Had we known that attractant, we could have eradicated it very quickly. The mango fly, after 3 years of attempting eradication, it just didn't work. Because it's a Q-lure fly. Q-lure is not as powerful as methyl. So it didn't work out. So that's a species that was not eradicated. So and Nauru never established, well, they established quarantine service, but they never implemented quarantine inspection. As a result, melon fly was reintroduced in 2001, and the interest and commitment faded. So now we have melon fly again, and we still have mango fruit fly. And now Nauru, because they no longer have phosphate, they get their income from maintaining detention facilities for immigrants to Australia. It's a pretty sad story. So, but the impact, 3 out of 4 species were eradicated. Over 30 national staff from the Pacific Islands were trained on fruit fly eradication, hands-on and quarantine surveillance. They formulated emergency response plans. And that paid off in the Cook Islands. Because of that training, they were able to eradicate fruit flies very promptly. We'll see that in a few minutes. And for the first time in their lives, children in Nauru are able to have a taste for fresh fruit. So French Polynesia in 1996, right after the training in Cairns, my colleague from French Polynesia, Rudolph Putoa, we spent a lot of time together in Cairns. We were among the minority of French speakers in that training. So we became very good friends. And Rudolph went back, brought back traps that were given to us to establish quarantine surveillance. And as soon as he went back to Tahiti, he actually detected a recent introduction of oriental fruit flies. So immediately, a few months later, they engaged an eradication procedure with using coconut husk blocks dipped in methyl and malathion, nailed to trees and dropped over the high mountains, these inaccessible terrain. It's very much like Kauai, the same topography, so dropped by helicopter, and some limited protein bait applications. So here was the number of flies per trap per day in traps, came to 40. So as soon as they applied the male annihilations, numbers dropped. But it took several years. So after 6 times every 2 months, after a whole year of block applications, the program was interrupted. No more funding. And that left small residual breeding populations of oriental fruit flies, very localized. So it's a little bit like you're sick, you take antibiotics, you take a few pills, you feel great, but there's still some left. So you can get even more sick. It's the same analogy. You have to keep applying the male annihilation until you're certain that it's eradicated. They didn't do that. So the flies started breeding again, and they kept trying to eradicate, and it didn't work out because again, the commitment had gone down. The first year there was lots of commitment, but program was running for too long. So in early 2002, they stopped the eradication. The flies started increasing in numbers. That's when Roger Vargas from USDA ARS, many of you know him really well, or used to know him. He got funding from USDA Foreign Agricultural Service to establish a parasitoid rearing facilities and do fruit surveys to introduce the parasitoid wasp that's established in Hawaii to control Oriental fruit fly, to introduce it to French Polynesia. So Roger and I and Neil Miller, who was working for Roger, all of us went to French Polynesia. I came from Fiji, where I was living, and Roger and Neil came from Hawaii, and we established a lab, and we established fruit fly and parasitoid wasp rearing. And I was there, and because I'm fluent in French, I helped a lot to as a facilitator between the French Polynesians, who many of them have limited understanding of English, and the American and Australian entomologists who speak their slang English are difficult to understand. So that was part of my role besides being the entomologist. So in early 2003, number of flies per trap per day reached 100. So late 2002, parasitoids were introduced, and there were 10 shipments, 500,000 flies released in a year and a half. And at some point, the parasitoids became established, started controlling, so the flies traps started decreasing. And here is long-term monitoring through fruit sampling. The number of fruit fly larvae per kilogram of fruits was 250 on guava and 130 in Tahitian chestnut, 250 in tropical almonds. So very high infestations. And the number of flies per kilogram of fruits over the years through the actions of the parasitoid wasps, 90% reductions in guava, 80% in Tahitian chestnut, 95% in tropical almonds. So the early success of biological control, the impact in Hawaii, was replicated in French Polynesia, has such a way that the number of flies per trap per day is now pretty low, and was reduced by almost 90%. And parasitism in fruits, over 70% of fruit fly larvae are parasitized. So Palau, the island of Palau, Oriental fruit fly was discovered in 1996. That's when I was working in Micronesia, and it caused extensive fruit fly damage. By that time, they didn't know until I gave a trap to an Australian who was traveling to Palau, and he just put a methyl-eugenol trap just for the hell of it and brought back the flies. And I saw, wow, these are not mango fruit flies. They're not the native fruit flies. That's Oriental fruit fly. So it had been established for several years already, but they didn't have any monitoring. So it was already large, well-established populations, so causing a lot of fruit damage. A socioeconomic study supported the $1.6 million eradication cost over 2 years, and the Palauans, well aware of the fruit fly problem, fully supportive. And they were going to do helicopter drops to eradicate. So, but the eradication They talked a lot about it for years, but it never came to be, was never implemented. So Oriental fruit fly is still in Palau, well established, threatening to spread to Guam, Yap, and Mariana Islands, any of these islands. And through DNA genetic studies, I determined that the Oriental fruit fly had been introduced from the Philippines. So I did with Harry Fay, Australian expert in fruit fly eradication. We did 3 missions in Palau to try to initiate area-wide control, but the commitment is very limited, so that has not happened yet. Cook Islands, well, in 2001, Queensland fruit fly was introduced from French Polynesia, because Queensland fruit fly has been in French Polynesia, introduced from Australia from New Caledonia since 1970. So because of the training in Nauru, because they had a network of surveillance traps, they detected right away. As soon as it became established, they detected right away, and they immediately started an emergency response, such that in less than 2 months, in less than 3 months, they were able to eradicate the fly. They did it with these male annihilation bait stations made of cardboard with fipronil, and they attached them to trees, and they did airdrops using an ultralight airplane with some bait sprays and crop sanitations. They destroyed 500 tons of contaminated mangoes. So it worked. Then 12 years later, 9 years ago, Oriental fruit fly was introduced to 2 islands of the Cook Islands from Tahiti, but they still had well-trained people who knew what they did and who were still had their good rapid response contingency plans, such that they were able to eradicate fruit flies from Rarotonga, a volcanic island, and Aitutaki, an atoll. And from May 2013 to August, so to April 2014. So to August, yeah, they were able in 3 months to eradicate, and they had some flies a few months the next year collected, so they continued the eradication, and they did it very, very seriously with this time with airplane drops and with coconut, so coconut husks trapped in fipronil and methyluginol and fruit destructions, as you can see. So they were able, so here a small Pacific Island nations program implemented by the Pacific Islanders that was successful. So it's a great achievement. So from these different programs I've been through, I've reviewed some of the lessons that we can take from that is if you have natural calamities such as drought and typhoons that reduce fruit fly populations, such as happened in Guam and Nauru, then it's the right time to hit and eradicate the fruit flies. It's the best time. So you take advantage of these natural calamities. It's good to have a good surveillance system with an active, maintained network of traps for early detections. That paid off in Auckland and Cook Islands. It did not pay off in Cairns. It took 3 years to detect the fruit flies. In Palau, they didn't have either, so I should have added Palau there. So there. And if you have a functional and effective entry and inter-island quarantine, such that they have in Australia and New Zealand, you can prevent most potential introductions. And Nauru did not have any quarantine inspections. A quarantine inspection in French Polynesia is not that effective. So they had these introductions. And you should be fully committed to carry the program to the end, not take shortcuts like in Nauru and French Polynesia, and not wait too long, too many years, like in Palau, that it's no longer practical to eradicate. Take no shortcuts. Don't do partial treatments or terminate a program early, assuming that the pest is eliminated. French Polynesia paid the big price for that. And apply correctly the protein bait spraying. There were some issues with the bait spray in Nauru and French Polynesia, which I did not go through, but it was not completely applied as it should. So you need to apply all the technology very correctly. And surveillance, early detection, and eradication is affordable to all countries, such as small nations like Cook Islands. So a lot of this work was published. So some examples of publications where you can read more about these programs. I take the Regional Fruit Fly Project in the Pacific, for which I worked from 1994 to 2002. And Alan Allwood was the chief technical advisor of the project. He's the greatest employer I've ever had. And I'm going to go see him in July in Australia. And Emma Vuetti, Fijian entomologist, I've worked with her for 8 years, and she lives now in Australia. I'm going to go see them in Brisbane, and it will be really great to see them again. Great thanks to Roger Vargas, who most of you, he's a legend in Hawaii. Unfortunately, he left us 4 years ago. So Roger came several times to the Pacific Islands, and we worked a lot together when I was still there. And Roger is the one who brought me from Fiji to Hawaii in 2003. And he's the one who organized for funding and convinced me to go back to do a PhD when I was in my 40s. That's when I was schooling with Roshan. We schooled together doing our PhDs. So we are brothers. And Ernie Harris, who implemented the first eradication program in Hawaii for fruit flies in the Mariana Islands. He continued working. He retired in 2006. He was 78 years old. And after retiring, he continued coming to the office and work on publications. And Ernie is the one who fine-tuned the mass rearing techniques for the parasitoids that we use in French Polynesia to control oriental fruit flies. So Ernie left us again 4 years ago. He was almost 90. So Roger and Ernie, they're my role models. So is Alan. They're the role model people I've really look up to as great inspirations. And I've been friends with them and their families ever since the days I met them. So Ernie, in 2016, he got the Congressional Gold Medal. So throughout his career, it was really hard for him in the 60s, 70s, and beyond, because he was a minority person, so he had issues, but he still managed to do really great work. And the full recognition, he got it at the end of his life with the Congressional Gold Medal. And he outlived most of the people who he worked with. So Ernie is my hero. So they're all are. And Emma is my sister. So these are my greatest fruit fly collaborators and colleagues. And just one last note, at the end of June or the end of July, I'm not sure, either one, there will be a book on the fruit flies of the Pacific Islands, fruit flies of Oceania, that that will be published. It will be about 200 pages, will be published under Insecta Mundi. I've been working for many years on that book. So it will have identification keys, color plates, and the entire host fruit records for every one of the 92 species of fruit flies in the Pacific Islands. And with that book, you'll be able to identify any of the fruit flies. So that will be my contribution to, my latest contribution to the fruit fly community. And that will be directly relevant to Hawaii. So I hope I was within time. Yeah, did a bit longer, but thank you very much for your attention.
Thank you, Luke.
For inviting me to this program. A good opportunity to see some of the friends after a long time, like Tracy and Luke. We haven't seen each other physically for a while, but at least on virtually we saw it today. Yeah, I will be talking on this Tamarixia production and biocontrol of Asian citrus psyllid. A little bit of my background, Roshan also mentioned. I came to Hawaii in 1997 and worked on what happened again? Worked on pineapple mealybug, its parasite-wide production with Marshall Johnson. And after finishing my PhD, I worked on that area-wide fruit fly control program, where Luke was also participating in the later half of the program. I left Hawaii in 2005 and came to California in 2010 to work on this Asian citrus psyllid. And since then, I'm working on this one particular insect, Tamarixia radiata. So today, I'll be talking about the ACP, HLB in a brief, and then HLB impacts, various control strategies. Biocontrol is only one of them. And then the current status of ACP and what we are doing on Tamarixia. So here, Asian citrus psyllid is invasive species. It invaded mainland USA in 1998 in Florida. And then it was reported from Hawaii in 2006, in California 2008, and other states like Georgia, South Carolina, Louisiana, and Texas in 2009. The insect needs this very young flush to lay the eggs. The adult can survive on older leaves, but does not lay any egg there. Eggs are laid only on the very young tissue. The nymph will feed there, and then the nymphs can feed on the mature leaves also. This insect alone is not the big problem. The biggest problem associated with this is it can acquire and transmit a disease, which is called Huanglong Bing these days, also known as green citrus greening. It is a bacterial disease, and so far there is no cure. So it is like once the insect transmits this disease to a tree, that is like a death sentence. Younger tree will die within like 5 years. Mature tree may live up to 10 years, but production will goes down drastically. So distribution of ACP and SLB in this map, I won't go in much detail. So it is distributed in most of the areas where citrus is grown, except a few places like Australia is still free of it. Europe is still like free of it, but most other areas like South America, North America, Asian countries, they are all invaded by this ACP and HLB. HLB transmission can occur by grafting if it is the graft tissue is taken from a diseased tree, but most commonly transmitted by ACP, the Asian citrus psyllid. Experimentally, Dodder, parasitic weed, was also able to transmit disease, but in nature, it plays very minimal role. HLB host plants, it is, all the citrus are susceptible. On the top of that, Murraya paniculata, it's a flowering plant, or it's a haze plant, live haze. It is also susceptible. But the curry leaf plant and Australian finger lime, they are resistant. So that is some role of these resistant varieties. I may talk a little bit later. So what is the impact of HLB? Before, so here in this graph, you can see the production of citrus in Florida, California, and other states like Texas and Arizona. Since this HLB was detected here in 2005, see how the production of citrus went down every year. And now it is almost as much as California. Here it was almost 3 times, or even more than that, when it was at its peak compared to California. California, the disease is not spread in the commercial orchards. It is found only in the backyard citrus. So the production has not been impacted. So we are very stable production in California. But if you look at Florida, there is a reduction so much. At 1 year, here, the California production was higher than Florida. It was because of the disease as well as the hurricane. What is the control tactic that California adopted? Number 1 is quarantine. As soon as the insect was detected, the selling of different seedlings was quarantined. The movement from insect detected area to the insect-free area was completely banned. And now there are diseases have been found. So even the transportation of fruit from one zone to another zone is restricted. So total area is divided into 6 zones. This red zone is where the HLB has been found. Other zones are HLB free. So there is a restriction on how they can transport fruit from this area to other areas. Chemical pesticides are used in the commercial orchard on a different forms of spray, like dormant spray, border spray, buffer zone spray. There are different names of the part of the IPM. I am involved in the biological control. And on the biocontrol, we have gone through all like 3 different aspects. Introduction, where foreign exploration in Pakistan, assuming that ACP is originated in that region of the world. And then we brought 2 primary parasitoids from Pakistan. There were some hyperparasitoids also in that shipment, but they were eliminated in the quarantine, and only the primary parasitoids were released. The conservation work is also going on, especially different resident predatory insects like green lacewing, different beetles. They are conserved, and they are very helpful in controlling ACP. And we are also working on some augmentative biocontrol, repeated release of tamaric specifically around the SLB find area and in some experimental area for the IPM area-wide ACP control. A fungal pathogen also have been evaluated, found effective, but it is not still easily available for the growers or the homeowners. On the biocontrol, predators, parasitoids, and pathogens, these are mostly the predators. Very important role. Most of them are conserved. Some of them can be grown in the insectary and also released augmentatively. Syrphids and green lacewings have been reported to have the greatest suppression ability, followed by the different ladybugs. This diomas is very specific to psyllids. And we are working on production of this. And there are some predatory mites and ants, but they don't play a major role as yet. The parasitoids that I'll be talking mostly, there are 2 parasitoids, this Tamarix radiata it is eulophyte. It is a solitary ectoparasitoid. Eggs are laid underneath the body of a nymph of ACP, mostly 4th or 5th instar, and then the larva stays outside and sucks the liquid from the nymph and kills it. Diaphoroncetus aligarensis, it is endoparasitoid, and it prefers to attack 2nd instar to 4th instar nymphs. The difference on the mummy you can see here is when Tamarixia, it makes a exit hole, it is in the anterior part of the body, and Diaphoroncetus usually exits from the posterior part of the body. So to introduce this, we went through the non-target test in the quarantine. We tested 6 different psyllid species, and among them, this potato psyllid was attacked by both the parasitoids, but less than 5%, because this is also a pest insect. And we reported that to USDA, and USDA still, though it was not very host specific as we wanted it to be, but we got a permit to release, and we released Tamarixia in 2011 and Diaphoroncetus in 2014. After that, we started producing this Tamarixia, and for some time, Diaphoroncetus also. Greenhouse production, we use now curry leaf plant in small cages and Asian citrus psyllid as insect host. And then the Tamarixia is collected manually with the help of a vacuum pump and then released. My involvement is mostly in this field cage production. Use this large trays. We use large cage to cover these trays, and we produce Tamarixia in the field cages. So our production process is we'll prune the tree and then cover it and introduce ACP from our greenhouse. The greenhouse colony of ACP is tested for HLB. It is free of HLB. So we grow HLB free ACP and release that. ACP multiplies in the cage, then we add Tamarixia radiata into that cage. And once it is pupating, we harvest it. We can use that cage one more time, or we clean it and move our cage to a next tree. So all the Tamarix they are collected, and we release is done all through the CDFA single window system. So all the releases through CDFA, my next door here is David Morgan's team, his lab. So they do the greenhouse production of Tamarixia as well as field release and monitoring. So far, more than 23 million wasps have been released in California. In the first phase, ACP, the Tamarixia was released wherever it was, the ACP was reported, finds. But later on, the ACP spread throughout Southern California. So in the second phase, there was a risk-based release, in the sense, if there is a higher risk of HLB or higher risk of ACP finding, that area got higher dose of Tamarixia. Where the risk was low, then it got lower risk of Tamarixia. But Tamarixia was released throughout, following a breed system. And in the third phase, we are now in the containment phase. What that means is wherever this HLB is reported. So most of the Tamarixia goes in that area. And some Tamarixia are also released along the border. We don't want any ACP invading from across the border here. So we release along the Mexico, California, US border here. And the new find sites, if there are some ACP reported in these sites other than the Southern California, in the Central Valley or in the other parts, we release there also, but very small amount of those are released there. And what we found is when the fruit is transported, it also transports ACP. So Tamarixia is also released along these fruit transport routes. So these are the highway. So we release along the highway. So here you can see how they are released. It is concentrated here in the LA basin. This is just a magnification of the same release sites. And so 72% of all the agents released in January went here in the HLB find area. So our focus now is wherever HLB is found, CDFA goes and applies pesticide in the near vicinity of the find sites, and the surrounding that site, we release Tamarixia so that the ACP production in that area is minimized, so it will not migrate to the production orchard. So here is the current status of ACP. These lines are the number of floss found on the trees. So flossing pattern remains the same. Here on this graph, you can see the population of ACP. Eggs here in the blue, then early instars of the ACP names, then late instars and adult. So they were pretty high in 15 and it started to go down every year. And there are like 50 sites being monitored every month. And now, sorry, very difficult to find in those monitoring sites. So ACP population has declined from very high to negligible here, though the floss development continues as normal. And the parasitization percentage, because there is no host, the parasitization percentage also has gone down, but it has helped a lot to suppress ACP in the urban area. So all of this, most of these releases are in the urban area, and we are monitoring in the urban area. Commercial area still depends on quarantine and chemical pesticide treatment. What we are doing now on the Tamarixia is we brought Tamarixia radiata from Pakistan. They were maintained as 16 different lines of T radiata. They were recombined and mass multiplied and released. After several years of releasing here in California, it is adopted, it is established in California. So our now new effort is to collect California lines of T radiata. And so far we have collected about 10 lines, and we'll be breeding that and multiplying it for field releases. So the original source was Pakistan, but now we are trying to establish our own California lines of Tamarixia and release them in the field. And so basically that is all of my presentation. It is a collaborative work, Citrus Research Board, USDA, CDFA, you see Riverside, Cal Poly Pomona, private insectary, and farmers. Everyone has contributed to help this program be a success. And as Rosen mentioned earlier, a couple of years ago, the DPR provided this whole team of ACP biocontrol a statewide recognition award. So that is all my presentation. Thank you very much for your attention.
Thank you, Rob.
Perfect. Thank you for the introduction. And hello, everyone. I'm happy to share some of my recent work with you today about parasitoid viruses and the opportunities they may present for future fruit fly biocontrol. So many of us are already familiar with parasitoid wasps, but for my talk, I just want to emphasize that this is a lethal interaction, meaning that once this wasp lays her egg into this caterpillar host, only one of those two insects will ultimately survive. Either the caterpillar will mount an effective immune response that kills the invading wasp egg, or that developing wasp will evade those host defenses and eventually kill the host. So this has led to co-evolution between parasitoids and their hosts, in which each is adapting to outdo the other. Arguably, one of the cooler adaptations that has repeatedly evolved in parasitoid lineages are these heritable associations with beneficial viruses, in which virus particles are produced in the reproductive organs of female wasps and then get injected into the host insect during oviposition. Inside of the host, the virus will infect host cells and manipulate host physiology in ways that benefit the developing wasp. So this can be through suppressing the host immune system or altering host development. Therefore, this can be seen as a mutualistic association because the wasp benefits from the activity of the virus while developing within the host, and the virus benefits from the replication and transmission supplied by the wasp. So parasitoids have repeatedly evolved these heritable associations with viruses. Shown on this parasitoid phylogeny here with little virus particles are several different independent acquisition events of viruses that we know of. During my PhD and now as a postdoctoral science, I've begun looking at the opine wasps, which primarily attack tifridid fruit flies and contain their own unique viruses. And our long-term goal here is to gain a more comprehensive understanding of these beneficial viruses by looking for new viral systems, as well as characterizing those that are understudied. So in efforts to discover new viral associations, I'll first mention this wasp, Phopius erisanus. This is a major biocontrol agent against fruit fly pests, which Luke had mentioned. And this wasp was not known to produce a virus until relatively recently when its genome was sequenced. And within the Phopius erisanus genome was found several clusters of these viral genes shown here in red. And so the presence of these viral genes suggests that this wasp may be producing virus particles. Next, the ovaries of female Phopius wasps were dissected out. And what we see here is kind of a similar picture to other parasitoid virus systems, which is at the end of the ovaries, we have this bulbous blue iridescent region known as a calyx region. In other systems, this is where virus particles are produced and stored, which is why they get that blue coloring. And so this suggests that Phopius also may be producing virus particles here. Further microscopy of the calyx region showed just that. It was found this dense array of these virus-like particles. We call them virus-like in this case because while they have a viral protein capsid, they don't have any viral DNA within them. So this virus was identified as an endogenous nudibvirus and represents an independent example of this phenomenon. So we're still at an early stage of understanding what these virus particles might be doing for Phopius wasps or what the virus might be doing within the fruit fly. That's part of the work that I have planned as a postdoc. But we're already seeing some features of convergent evolution between this system and other parasitoid viruses, which is pretty cool. For my dissertation, I worked on another opine wasp shown here, Diachasmomorpholongicodata, or D. longi for short. And my dissertation had the overarching goal of characterizing a divergent viral association. So this wasp also lays its eggs into tifridid fruit flies. In my former lab, we reared our D. longi on this fly here, Anastropha suspensa, the Caribbean fruit fly. Yeah, that's right. And in this system, the virus is not produced in the ovaries of female wasps, but rather in the venom gland, which is associated with the ovaries as part of the female reproductive system. And as you can see in this instance, it's the venom gland that has a natural blue iridescence under the microscope, which indicates all of the virus particles that are stored within it. This virus is a pox virus known as DLEPV for short, and pox viruses are very different in their basic biology than other parasitoid viruses, making this an ideal system really to investigate. As part of my PhD, we have been able to experimentally remove the viral population from D. longi wasps using RNA interference technology. So in this image is a normal venom gland up top, which has that bright blue iridescence. Some of it's actually leaching out of a ruptured tubule. But in a viral knockdown wasp venom gland, it's relatively empty and does not contain the virus anymore. Using those wasps that are now devoid of the virus, we measured parasitism success of the wasps. And compared to those that have the virus, which survive at really good rates overall, when the wasps don't have the virus, that emergence rate plummets. And so this tells us that the virus is heavily beneficial for the wasps to have. So one of the projects from my PhD that I'll be sharing with you today looked at the potential role of DLEPV, this symbiotic virus, in potentially shaping the host range of its parasitoid. So while I was still a grad student back at University of Georgia, I started this project in 2019 by coming to PBARC here for the first time. And I was here for 3 months to conduct this research project. While I was here, I was interested in a unique aspect of D. longi ecology, which is that it's a generalist parasitoid species, meaning not only can this wasp successfully develop within Anastropha suspensa flies, but it can develop within a number of different fruit fly species belonging to a number of different genera. Because of this generalist behavior, D. longi, along with dozens of parasitoids, as Luke had mentioned earlier, were released across the Hawaiian Islands to try to combat pestiferous fruit fly species. And so 3 of the big ones here are shown, the medfly, the oriental fruit fly, and the melon fly. And D. longi, along with Phopius erisanus wasps, have actually led to efficient suppression of both medfly and oriental fruit fly populations here. However, there's no data suggesting that D. longi uses melon fly as a host. So the first thing I did was to measure parasitism success of D. longi within these different fruit fly species. And what I found is that in both medfly and oriental fruit fly, the wasps emerge at relatively good rates. However, not a single wasp ever emerged after using the melon fly as a host. What this tells us is that both medfly and oriental fruit fly are permissive hosts for D. longi development, while the melon fly is a non-permissive host, because even though the wasps will lay eggs into melon fly larvae, those eggs never survive to adulthood. Naturally, I was interested in whether DLEPV, the virus, is somehow involved in maintaining this unique host range pattern. So next, I measured the abundance of this virus throughout parasitism within each of the different fly species. And what I found is in both medfly and oriental fruit fly, we see significant increases in viral abundance over time, indicating that the virus is able to replicate in both of these fly species. However, we see no increase. In fact, we see a significant drop in viral abundance within the melon fly, telling us that the virus cannot replicate within melon flies. And so this shows us that there seems to be a link between the ability of the virus to replicate within these different fly species and the ability of the parasitoid to successfully use these flies as hosts. To get a better resolution of the activity of this virus while within the different fly species, I purified virus from the venom glands of female wasps and then injected that virus into the different flies at 3 different doses. So first, a physiological dose. This is the amount a wasp would normally inject during oviposition, followed by 2 different dilutions of that physiological dose. First, I simply measured whether flies could survive viral infection at these different doses. So each of these pie charts here indicates the relative proportion of flies that emerged as adults after being injected with the virus as larvae. And in general, what we found is a gradation of susceptibility to DLEPV by these different flies, where up top with the medfly at both the highest 2 doses, absolutely no flies emerged. And only 11% of flies emerged at the lowest dose of virus. Then with the oriental fruit fly, again, no flies are surviving that highest dose of virus, but we do see at the intermediate dose, a small proportion can handle the virus, and then almost half of flies at the lowest dose can survive. Lastly, with the melon fly, right away, we find a small proportion of flies can survive that highest dose of virus, followed by increasing proportions that can survive the lower 2 doses. And so again, with the melon fly is by far the least susceptible to viral infection, while the medfly is the most. And these patterns are again linked with the ability of the parasitoid to use these flies as hosts. Next, I measured viral abundance after injection with the virus at the same 3 doses within the different flies. And generally, the mortality patterns that I just showed you are mirrored by the ability of the virus to replicate at those different doses. For example, in both medfly and oriental fruit fly, where we see doses in which there's good virus replication curves, that is always correlated with doses in which no flies are surviving viral infection. However, with the melon fly, when all 3 doses lead to significant drops in viral abundance, that's always correlated with some proportion of flies that survive all of the doses. Lastly, I've just begun analyzing a large transcriptome data set, looking at viral gene expression during the initial 24 hours of infection. And so this heat map here shows the relative expression levels of all 200 viral genes. And what I want you to take away is basically that there's this huge die-off of viral gene expression within the melon fly that we don't really see in the other two fly species, telling us that the virus seems to be able to infect melon fly cells and start the replication cycle, but is pretty quickly just breaks down, which is probably why the melon flies are able to survive. So in summary, D. longi displays this varying compatibility pattern within these tropical fruit fly species, where both medfly and oriental fruit fly are permissive hosts for D. longi development, while the melon fly is not. And secondly, the activity of DLEPV, the symbiotic virus, is strongly associated with this host compatibility pattern, because in both the permissive species, we see rapid virus replication and abundant fly mortality, whereas in the non-permissive species, we see no virus replication and much reduced mortality. Taken together, these results tell us that DLEPV could be a major contributor to the success of this parasitoid as a generalist and more broadly as a widely released biocontrol agent. Next, I want to share some very recent preliminary results looking at how frequent this symbiotic virus is in wild populations of D. longi here on the Big Island. So this has been an outstanding question for a while now, which is that we clearly see in the lab that this virus is super critical for the parasitoids to keep, because when they don't have the virus, their parasitism success is basically nothing. However, there's no data yet looking at whether this virus is also important for wasps in the wild. And so to start getting at this question, we tried to sample wild wasps. And the main way we did that was to collect fruit that we knew would be infested with fruit flies, and then try to see if any parasitoids also popped out of those collections. So we started here locally in Hilo at OK Farms, which among many things, they grow coffee. And the coffee cherries here at OK Farms are normally known to be infested with medflies. So last fall, on 4 different weeks, we sampled coffee from the farm and we took it back to the lab. And after a certain amount of time, any flies that were developing within the coffee cherries had exited the fruit to start the pupal stage of their development. And then when they were pupae, we could individually place pupae into individual wells of these plastic grid structures, which allowed us to then monitor ultimately what came out of those pupal cases, whether it was an adult fly or maybe an adult parasitoid. And due to the bright orange coloring of D. longi, we could pretty easily pick out the wasps as they emerged. And we were specifically trying to collect the females because that's where the virus is maintained. So we took our wild D. longi, and the first thing we did was to dissect them and look at their venom gland, which is a really quick way to sort of assess whether the wasp may be infected with this virus, whether we see that blue iridescence of the venom gland under the scope. And so we did see some that looked just like this venom gland, but weirdly, we also saw wasps that had a venom gland that looked like this, which in general shares the same overall morphology of the one I just showed you. But in this case, the accessory tubules are much, much shorter. It seems that they're pretty truncated compared to the other type. And these venom glands were kind of clear under the microscope. It did not look like they were infected with the virus. So this difference in venom gland morphology was kind of puzzling to us at first. But this image I'm showing you is actually taken from a D. longi wasp collected in Thailand. And after doing some more reading, I actually learned that in Thailand, D. longi is a species complex. It's not an individual species, but rather at least 3 to maybe 5, I think, cryptic species. So this phylogeny shows distinctly wasps falling into 3 different species groups in Thailand. And researchers have shown that these species are reproductively isolated from one another. They do not produce fertile offspring. However, they are very hard to distinguish from outer morphology and therefore are cryptic species. So I was interested in whether that might be the case with the wasps we had collected. And so I extracted DNA from each individual wasp and then sequenced part of the CO1 gene, basically used to DNA barcode the wasps. And this alignment here shows the sequence for each individual wasp stacked up. And what we found is basically two main groups that highlighted our different positions along the sequence where the two groups differ from one another. And overall, these two groups really only shared about 93% DNA similarity, which is a much lower percentage than what we normally use to consider individuals to belong to the same species, suggesting that these are in fact two different species. Next, I built this phylogeny, which confirmed those results. Here are individual D. longi clustered into two distinct species groups, and they're correlated with that difference in venom gland morphology that I showed you before. So these are definitely two distinct species. Also within this tree, I've included the colony of D. longi that we maintain here at PBARC. That falls within the first group. I also included some of the Thailand wasp sequences, which both cluster separately as we would expect. Yeah, so this was kind of surprising, but cool to see that we have these different species complex members floating around here. Next, we screened each of these individuals for the virus using PCR. And what we found is the top group, the top species of D. longi, 100% of individuals were infected with the virus. And in the bottom group, only 22% of individuals screened positive. And to be honest, I'm not convinced that these individuals down here were actually infected. I think maybe they were contaminated because these two groups of wasps are attacking the same fruit fly hosts, and therefore you might get some virus transfer between individuals. But regardless, the top group is definitely stably infected with the symbiotic virus, which was neat to see. So in summary, D. longi represents at least two cryptic species here on the Big Island, which is supported by DNA barcode differences, phylogenetic analysis, and differences in that venom gland morphology that I showed. And secondly, one of these two species is consistently infected with DLEPV, which tells us that similar to what we see in the lab, a viral infection is really important for this species to have in the wild. Lastly, I think these results are kind of exciting because they suggest that the acquisition of this virus may be correlated with the speciation of the D. longi lineage. Because we find the virus in only one of the species, and it's correlated with that expansion really of the venom gland, which is where the virus is stored. So I think it'd be really cool to get a colony of the short venom gland wasps going here so that we could perform comparative experiments between the two species, and from an evolutionary standpoint, maybe get at how this virus was acquired by the wasp in the first place. So I'm definitely planning to expand this study by sampling more wild wasps here in Hawaii, and maybe also in other parts of the world where D. longi is established, to kind of grow this data set and look deeply at the species complex aspect as well as how the virus kind of fits in. Lastly, I want to wrap up today by briefly discussing some of the prospects I think this kind of work can have in future fruit fly biocontrol endeavors. My PhD work was very much from an evolutionary biology standpoint, but now as a postdoc with the ARS, I'm excited to look at this work in an agricultural context. As I've mentioned today, these two parasitoid species, which we have colonies of here, as well as they're established on the islands, are widely successful biocontrol agents against fruit fly pests. But only really recently did we know that both of these parasitoids harbor these heritable viruses. And so these viruses represent previously hidden aspects of natural biological systems that are used to target fruit flies. And as I've shown you today, at least one of these viruses is highly lethal to a number of different fruit fly pests. So part of the work I have planned is to similarly characterize the function of the virus-like particles in Phopius erisanus, but my guess is that they are similarly manipulating host physiology, although maybe through a different mechanism. In order to use these viruses and future biocontrol efforts, we really need to understand the molecular basis for the effects they have on the flies. And one way to do this would be to identify the specific viral genes that are causing the phenotypes that we see. So for example, with DLEPV, we have sequenced the viral genome, and we can use that list of genes and try to narrow it down to only those that are killing the flies, basically. Luckily, we're well positioned here at PBARC to start this kind of work by combining state-of-the-art techniques in functional genomics and manipulative genetics. For example, we can use RNA interference, which I have now developed in both D. longi and Phopius erisanus wasps, to knock down specific viral gene expression. And we can couple that with things like CRISPR genome editing, which have been developed for a number of different fruit fly species here. And together, hopefully we can use those tools to really identify those specific viral effector genes or proteins that are killing the flies. Later down the road, once we've identified these viral effector proteins, and after we pass a million regulatory hurdles, we could eventually transition into developing these as bioinsecticides. So one example for this would be to genetically engineer plants to express these viral proteins so that when the fruits become infested with flies, those flies will consume the viral proteins and hopefully die, leading to population suppression. But that's just one idea. I think there's a lot of potential now that we know about these fruit fly targeting viruses to either exploit the viruses themselves or specific viral genes in future biocontrol innovations. An additional benefit to studying these molecular dynamics is that it could help inform the selection of future biocontrol agents by identifying molecular traits like heritable viruses that are shared by very successful parasitoids. So for example, out of the dozens of parasitoid species that were released into Hawaii to combat fruit flies, why is it that these two are the most dominant ones today? And it very well could be that they have these unique viruses that are helping them out. But just studying these sort of shared common molecular traits from successful biocontrol agents could then be, applied to completely different pest systems. And lastly, there's additional uses for understanding these virus-fly interactions, such as identifying the physiological pathways that the viruses are targeting within the flies that can later be exploited with other insecticides. So overall, I think this is a really fascinating avenue of research, and I'm happy to be a part of it. I would like to thank all of those at PBARC who have helped me with these research projects so far. Especially Scott Guide, my postdoctoral advisor. I'd also like to thank our collaborators on both the D. longi and Phopius projects, notably Galen Burke, my former PhD advisor. And lastly, I would like to thank Janae Johnson, who took those beautiful insect photos I showed. And thanks for your attention.
Also see my slides, correct?
Yes.
Okay, thank you. Well, good morning, everyone, and thank you very much, Roshan, for this opportunity, and to the other speakers today. I thoroughly enjoyed your talks. Thank you. And I'll be changing this morning's topic a bit as I'll be presenting about an app, the My IPM Hawaii app, and not about research. And I just want to clarify that although I am the only one presenting today, the development of this app would not have been made possible without the assistance and contributions of several amazing collaborators whose names will be mentioned later in the presentation. But first, let me give you a bit of a background about these apps. There are two other My IPM apps available. One is focused on the insect pests and diseases of fruit and tree nut production in the eastern US, including peach, apple, grape, cherry, strawberry, and pecan. And this is the first version of the app. And I had the opportunity to provide contents for this app before coming to Hawaii. And the second one, at the bottom here, was produced primarily for disseminating IPM information of row crops, including corn, peanut, soybean, cotton, and sorghum. So why were these apps made? First, provide growers, users, stakeholders, and agents with relevant and up-to-date information anytime and anywhere. Integrate pest diagnostics, spray guide info, fact sheet info, as well as resistance management recommendations. and simplify information with interactive tables, pictures, and relevant links based on the profiled crops or pests. And fourth, provide a handy resource for our stakeholders regarding correct approaches and diagnostics. Recognizing the usefulness of the apps to the respective agricultural systems in the mainland, we thought it'll be useful to develop one that caters to the Hawaiian growers and stakeholders as well. And hence, the My IPM app Hawaii version has been developed. It is primarily designed to provide integrated pest management information for important crops and business sectors relevant to the state of Hawaii. Currently, we have the following information on insect pests of ornamentals, including anthurium and orchid, fruit trees, including avocado, papaya, and banana, and vegetables as well. We have information on coffee and macadamia, and we also have recommendations on post-harvest quarantine treatments as well. And there will be more to be added in the near future, including sections on urban pest, coffee and macadamia diseases, vegetables, beneficial insects, and invasive pest watch list, which I believe would be of interest to this group. This app was made possible by the cooperation and collaborations by these amazing individuals, as I've mentioned earlier. Our current active contributors for ornamentals are Joanna Blousey and Cindy McCarty. For avocado, we have banana, coffee, and papaya are Mark Wright, David Hansberger, and Maya Hansberger. For cabbage, it's Rosemary Gutierrez as a extension agent in Maui. And Roshan here has contributed to content on papaya and sweet potatoes. And Kunui's materials were also used in the app. And more contributors will be coming on board soon as well, including Lisa Keith and Roxana Myers, who will be putting out materials on macadamia and coffee diseases. An urban pest section will be handled by Reina Tong and Dr. J.Wei Tai. And a few UH graduate students as well have expressed interest to help us with content contribution, including Kevin Armstrong, Christian Strait, and Ellie Nyarkiano. And the development, support, and programming of this app are handled by the folks associated with the UGA, University of Georgia, Clemson University, and Southern Region IPM Center. And the development and maintenance of this app is funded by USDA NIFA via the Southern IPM Center. So where are we getting the information from? The information that we put out in this app are obtained from various reputable sources, including the Crop Knowledge Master, which is a website hosted by UH Extension that contains information and pest management of Hawaii grown crops. We also base some information from publications by the University of Hawaii and the College of Tropical Agriculture and Human Resources. We also referred to data published in original research and review journal articles as well. The chemical pesticide information that we put out are taken from the crop data management systems and by consulting the pesticide labels as well. And we use a lot of images in this app, which came from the authors themselves, Bogwood Images, and also from CTAR. So how do you download this app? How do you get access to this app? Thankfully, the My IPM Hawaii app is available for free. How good is that? So it's available for iOS and Android mobile systems. Take note, this is not a web-based or desktop-based app. It is only available for phones. So you can simply search My IPM Hawaii with spaces in the Apple App Store or the Google Play Store. And as an example here, using an iPhone, you can type My IPM in the search box. Once the app is shown, you can tap on the icon here on the upper right to download, and voila, you should see the app on your home screen by then. And important to note that you need internet connection to search and download. And now to open the app, just tap on the icon and it's ready for use. And yes, it looks empty at first glance, but wait, you can tap on the cloud icon again on the upper right hand of the screen to see the list of available sections for downloading. And here is our current list. There are sections in different crops, as I've mentioned earlier, and their corresponding pests, as well as a section in post-harvest quarantine treatment. The list is arranged alphabetically. The app also allows users to choose and download the sections of your interests by tapping on those sections. This is a really useful feature of the app. For example, if a grower only grows macadamia, he or she may not need to download the other sections. On the other hand, if a grower grows a lot of crops, he or she has the option to download all applicable sections. Check marks, by the way, indicate the choices for download. And then after making a choice, you can tap update on the bottom to download the chosen sections. For iPhone users, you can see this notification here, which just means you have to confirm the download by tapping on the download button. This is unique to iOS. Android phones do not require this step. And now the chosen section should be visible on the app's main menu, as you can see here. This app also offers the flexibility to change your crops or sections at any time by simply repeating the same process. Whenever updates are available, users will be notified and given the choice to upload now or later. Updating the app requires internet connection, and to note, if anything is not working and after an update, unfortunately, deleting and reinstalling the app would help solve that problem. Once information and updates have been downloaded on the phone, the app can now be used offline. This is very helpful, especially in areas where cell phone signals are spotty or when a user does not have access to cellular internet data in the field. And now we'll explore the features in each section by tapping on the macadamia nut section. Once you're in, there are two ways to find specific entries under each section. For the crop and pest section, you can swipe the photos left or right to find a specific pest that you're looking for. This is good to use if the user does not know the name of the pest, but he or she is familiar with what the organism looks like. The other way to find entries is by simply tapping on the select button on the upper right side of the screen to access the list view. In this view, all the insect or disease pest profiled for each crop is listed alphabetically. This is good to use if the user knows the name of the organism. So let's click on one entry, and in this example, it will be the macadamia felted cockseed, which is an invasive species of macadamia. And this is what you will find after tapping on one pest. Tapping on overview, you can find descriptions on pest biology, distribution, damage, and management. On the bottom here, you can tap on gallery to view the zoomable photos relevant to the pest that is being profiled. Under chemical control tab, a description of available control options is also provided. And one thing that the Hawaii team was able to push for this app is the inclusion of the non-chemical control tab that contains a summary of non-chemical based control tactics, including cultural and biological control. And this is an IPM focused app after all, so we have to make sure to highlight other control tactics as well. So this app also features interactive tables under active ingredients and trade name tabs featuring available conventional and organic spray materials. By swiping the columns on the table, left or right, you can also see other information such as rates per acre, pre-harvest intervals, and re-entry intervals, among other things. And now moving on to the post-harvest quarantine treatment section, the presentation of information is a bit different in this section because instead of profiling the pest, the profile entries contain the treatment recommendation for specific crops. In this section, swiping the photos of the crop left or right allows you to access the information for each crop. However, it is not the fastest way of doing it. I personally would recommend using the list view by tapping on the select button here to access the list of crops. As you can see here, the post-harvest treatment recommendations are available for numerous crops listed. Thanks to Peter Follett, the list is in alphabetical order. By tapping on each one, you'll see the detailed information. So let's click on dragon fruit here. In this example, we have dragon fruit in the picture with the embedded information on irradiation recommendations. And by tapping on Overview tab, you can also see the treatment description, which is similar to what is actually placed with a picture, but it would be bigger if you would not be able to see the font. This particular feature is quite unique to the Hawaii version of this app. The information we are providing in this app is continually updated by UH Extension specialists, agents, and graduate students, and USDA ERAS research scientists. And this can actually be found on the About section in the app. And we can change the content and push to publish anytime using the authoring tool. For those of you who would like to become a contributor or explore the possibility of using this app to share information to your stakeholders, I'd be happy to discuss the details with you. It is a bit of a straightforward process and user-friendly as well. And now, if we still have time, Roshan, I'll be glad to do a short demo of the app. If we do not have time, however,
Yes,
feel free
yes,
to
sure,
contact
yeah,
me. Is
yeah,
it okay?
sure, yeah.
Okay, all
I
right. So,
extended this for 12:15.
okay,
Yeah,
great.
I just messaged, yes.
Oh, okay, thank
Please.
you.
Mhm.
Let's see, I will be sharing screen. Start broadcast. 2, 1. All right, can you see my screen? And
Yes.
okay, yes, okay. All right, so I have the 3 apps installed in my phone right now. And assuming that you were able to actually download the My IPM app here, as you can see in my screen, you can tap on My IPM Hawaii, which unfortunately, you can see my finger as I'm tapping on my screen, but I just did that. I tap on the icon. And like I said earlier, it looks empty at first, right? So don't worry, you can tap on the upper right-hand corner on that cloud icon right there. And as you tap on it, you can see the list of entries that we have right now, or sections that we have right now. So I'm biased on Macadamia Nut because that's my system that I'm working on right now. So I'll tap on Macadamia. And for me, for you to actually have that check mark, you just easily just tap on Macadamia. And then once you decide which sections you would want, you can just tap on update. It is updating. It may take some time depending on how big the section is. And you would see, if you're using an iPhone, you would see this notification, and it's just easy. Tap on download. Okay, it's downloading right now. Right, so in this process, I'm connected to internet. So it is important to be connected to the internet at this point. And going back to the main menu, for some reason, technical difficulty. I stopped sharing for some reason. Okay, it should be up again. Okay. Okay, we're back here. All right. So going back to the main menu, there is the section that I chose to upload or update earlier or download. So I can tap on this section, and this is what I would see first. And the list of the organisms or pests are arranged alphabetically. In my section, the male coacidum is the first on my list. You can choose to swipe left or right. So my next one, when I swipe to the left, I have MFC. If I swipe to the right, I have TNB and so on. So this is good if you know what you're, if you're not actually quite familiar with the name of the insect that you're looking for. But the other option for you is, if you know already what the common name of the insect or disease, you can click on the upper right-hand corner, select, and there you would see a list, the list of pests that have been profiled, okay? And going back to this, let's choose MFC again. We can click on overview, which contains all the information that are relevant for the users to know about this pest, common name, scientific name, distribution, damage, and so on. And clicking on the gallery on the bottom here gives you access to the photos that are relevant to the pest that we have profiled. And I can click on this one here, and I can zoom it in and out, and so on. Okay, and we try to be as detailed as possible, so we actually do include captions in our pictures too. All right, so going back again to this main menu for each of the pests. As you can see here, you can click on chemical control. There is a summary of what we can use for this pest in terms of chemical control. And you click on active ingredients. They do not have a lot of options, but it does contain active ingredient, IRA code if you want to be able to actually make sure that they take into consideration insecticide resistance management. And we have rate per acre, PHI, REI, and so forth. And one thing that you would actually want to know as well is, aside from the conventional control, we also have the organic information of materials that growers can use too. And going back here, as I've mentioned earlier, we were able to push for the non-chemical control tab to be placed in the main menu. So this leads you directly to the other control strategies aside from the chemical options. All right. And if you want to know about the people who are actively contributing to this section and you want to kind of send them a message, you can go to About My IPM. And these are the names. And we will be updating this as we actually add more sections and we have more contributors to be helping us. And if you would like to give us feedback, we do have that option as well, which actually would lead directly to me and to the other person who's helping us on the programming side. All right, so that's pretty much it. And if I can also briefly show you the post-harvest quarantine treatments, because I focused on that earlier. All right, so let's see if there's a lot of photos here for this section. Oops, it's 22 MB. So, all right, so Peter Follett publishes a summary of the information that I've actually put in here, but we made it into a format that is mobile friendly. So there's a summary of things that of the list that we have here, and we can just swipe right or left, and it's alphabetically arranged. You have atemoya, avocado, you have banana, and as you can see here, there's a bit of a description as to what the recommended post-harvest quarantine treatments are available for this particular crop. And if it's too small for you to read, you can click an overview here, and it's essentially the same information. And like I said earlier, this might not be the easiest or the fastest way to find the crop that you're looking for. You can click on select on the upper right-hand corner, and it'll give you the list of all the crops that we have profiled under this section. All right, so if you have questions or want me to show something else that this app can use, can offer, just feel free to let me know. Thank you.